Hello everyone and welcome back to microbiology. Today we'll be doing a review for Unit 1 exam. Most of it will be a sort of a game where I provide a term, a definition, or a concept, a method, something like that. And then it's your job to try and think of as many little details as you can about that. And then in the next slide, I'll show some details about whatever it was that was the particular question. We'll go through many of those slides, and then at the end, we'll do a little bit of a review on the math. So I've posted the math problems PowerPoint that we did earlier within this lecture, and we'll go through them one by one just to make sure you're prepared. So as an example, the first question would be, or term would be a primary stain. And here I would pause and allow you to collect your thoughts and try and think of some ways to define that and things that are associated with it. And then after a period of time, I'll move to the next slide. And here we have, the primary stain is the first stain in a differential stain. Here in gram stain, it's crystal violet, and acid-fast stain, it's carbonyl fusion. It's the first stain that's applied, usually followed by a mordant, and then a decolorizer, and then a secondary stain to contrast that from the first stain. So let's begin with number one, and that will be Streptobacillus. Okay, so Streptobacillus is a type of bacterial morphology. It is a bacillus means that it is rod-like in shape, and strepto means that it's made of a long chain arrangement. All right, number two, Psychrophile. Okay, so psychrophile is a way to define microbes depending on what temperature they grow at. In particular, psychrophiles have an optimum growth temperature of around 10 degrees Celsius, and they can grow below freezing and roughly up to about 20 degrees Celsius. And remember, for the exam, I'm not going to test you about each one of those little interfaces where you might cross from psychrophile to mesophile. I don't care if you know that mesophile ends at about 15 degrees temperature and psychrophile ends at about 20 degrees Celsius in temperature. But I give you something that's solidly within one of these groups of temperatures. You should be able to tell me what group of microbes that is. So if I say 90 degrees Celsius, you should easily be able to tell me, oh, that would be a hyperthermophile. Okay, number three, Archaea. All right. So there are a lot of things that you should know about Archaea. They are prokaryotic, single-celled. They do asexual reproduction. They have no peptidoglycan in their cell walls. They can live in extreme environments. There are three types: the methanogens, and that means that they generate methane; thermophiles, and that means that they love warm conditions; and halophiles, which are microbes that can live in very salty environments. Number four, parts of a microscope. In particular, we're talking about light microscopes here. Okay, here we go. There are many components to the light microscope, and the exam might have a diagram somewhat similar to this, and you might need to be able to fill in the blanks. So we can do this many ways. We can start from the bottom where we have our focusing knobs. There's always a fine focusing knob and a coarse focusing knob. The coarse focusing knob, remember, is usually used to initially find the object, and then the fine focusing knobs are used for fine tuning, for increasing the focus. We have the base where everything sort of sits upon. We have the illuminator, which is the source of the light. We have the diaphragm, which controls the amount of light that enters the condenser. Above that is the condenser, and the condenser does exactly what it says. It focuses the light through the specimen. Next is the stage where the specimen is placed on, so the glass slide that you would fix your specimen to. Above that is the objective lenses, and the objective lenses are the first lenses that help magnify the image. The objective lenses go up through the body tube and are reflected onto the ocular lenses, which are the eyepieces, and the eyepieces do the final magnification of the image. There is also the arm, which is that kind of that back piece that connects the base up to the top where the ocular lenses and objective lenses are located. Okay, number 5, refractive index. Okay, the refractive index basically describes the light that bends as it passes through a material. And we need to be aware that there are different refractive indexes depending on the material. So air and glass are quite dissimilar in their refractive index. And as you can see in this image, as the light reaches the glass slide, if there is nothing but air between that and the objective lens, you tend to have a higher degree of refraction or bending of light in the air. And for samples where you have really high magnification, you need to direct that light more correctly into the objective lens. In order to do that, you will use a immersion oil, and that immersion oil makes contact with both the glass slide, which is where the specimen is found, and the objective lens. And therefore, as you can see with that blue line, the light is not refracted as much, and it actually makes it to the objective lens and eventually to your eyes. Okay, number 6, acid dye. Okay, there's a lot that we could unpack here. So acid dyes are components of stains, and stains, as you know, are helped to visualize microbes under a microscope. The component of the stain that contributes to color is called the chromophore, and this chromophore can be positive or negative. A negatively charged chromophore is an acidic dye, and a positively charged chromophore is a basic dye. So these acidic dyes are typically used to stain the background. And that's because most microbes also have a negative charge on the outside of their cell. And so these two negative charges repel each other, just like two negative sides of a magnet. Well, in a sense, similar to that. Likes repel each other. And so the negative chromophore can attach to the slide, but doesn't attach to the cell itself. Number 7, acid-fast stain. Okay, so this one might be a little bit tricky because we just discussed acidic dyes. But an acid-fast stain is used to stain the genus Mycobacterium, which are resistant to acid alcohols, which are used as a decolorizer. And that's due to their mycolic acids. As you might remember, they have mycolic acids in their cell walls. The process is like we see here in this image. First, we apply our primary stain, the carbonyl fusion, and then we then apply that, we put that slide under heat, usually a moist heat so it doesn't get too dry. And this is serves as a mordant for that carbonyl fusion. And then we expose cells to acid alcohol, and the Mycobacterium are resistant to this acid alcohol, hence they're acid fast, whereas other cells may not be. So then post decolorizer, methylene blue is added as the counter stain, and so non-acid fast bacterium will appear as purple, and acid fast bacterium will appear as sort of this pinkish reddish color. Now we have Darkfield Microscopy. Okay, so the Darkfield Microscopy uses a special condenser. Remember, that's the part of the microscope that allows that kind of concentrates the light to the image. And what it does is it blocks out most of the light. And all the light that you can see is the light that gets diffracted by the specimen against a dark background. So that's why we have that name, Darkfield Microscopy. It's often used for specimens that are difficult to see using bright field microscopy and don't stain well. So in this example, we have Paramecium, a eukaryote. Number 9, Robert Koch. Okay, Koch was a very important figure in the history of microbiology. He was the first person to connect a specific bacterial species, Bacillus anthracis, to a specific disease, anthrax. He developed Koch's postulates, which was a series of experiments to determine if a disease was linked to a particular pathogen or not. And he was very important in the development of the germ theory of disease. Number 10, Lophotrichous flagella. Okay, so Lophotrichous, it means that we have a tuft of flagella, and we can have a Lophotrichous polar flagellum. So here we have it at one end. It could be any of these other forms as well, though. Next is the slime layer. Oops, sorry about that. We'll give you another second here. Okay, so the slime layer is a type of glycocalyx. It is made of unorganized material that is loosely attached to the cell wall. If it is well organized and attached firmly to the cell wall, we refer to it as a capsule. And the slime layer allows for cells to attach to each other and to surfaces and to form biofilms. Okay, number 12, gram-positive cell wall. Okay, so gram-positive cell walls, they are rigid. They have many layers of peptidoglycan. They have these teichoic acids, which are the black components here sticking out of the cell. These peptidoglycan cell walls on the outside of the membrane, and they are quite susceptible to antibiotics that attack peptidoglycan, whether breaking it down directly or its formation. In this slide, I also show gram-negative cell walls, and they are not as rigid. They're made of just a few or one layer of peptidoglycans. There's no teichoic acids. There is LPS. The peptidoglycan is located between the membranes, and it is not susceptible or much less susceptible to attack on the peptidoglycan. Fomite. And the fomite is any non-living object that may be contaminated with microbes. Number 14, sanitization. So remember that sanitization is not sterilization. It is used to reduce the number of microbes on fomites. It doesn't destroy endospores, so we don't get true sterilization. And it usually involves a relatively short exposure to a sanitant, but still meets public health standards. 15. Pasteurization. Pasteurization is a brief high heat exposure to food products such as milk, dairy, juice, sometimes eggs and honey, in order to destroy pathogenic and spoilage microbes. It does not sterilize the food, and it's used in order to help preserve the flavor and nutritional profile. We have two main types, which is high temperature short time, which is 72 degrees Celsius for 15 seconds, and ultra high temperature at 138 degrees Celsius and 2 or more seconds. This is going to be a lot like the question about the groups of microbes that grow at different temperatures. I won't require you to memorize the exact temperatures here, but if I was to provide you with temperature and time, you should be able to tell me which version of the two it is. Okay, 16. Fungus. All right, fungi are eukaryotes. They're single-celled or multicellular. They can do sexual or asexual reproduction. They are heterotrophic. Their cell walls can contain chitin, and two examples of them are molds and yeasts. Okay, 17, standard plate count. The standard plate count is a method for determining the number of cells within any given solution or culture. The way this works is we start with an original inoculum. This could be your culture that you grew up. And you must go through what we call serial dilutions. So in this example, we're adding 1 ml to a 9 ml broth, and that makes a dilution of 1 to 10. 1 being the amount added, and then 10 being the total new volume. So that's 2 serial dilutions we see here. 1 ml into one broth tube. You mix it, take 1 ml and put it into another one. And you do a series of serial plating in order to try and reach a number of cells on a plate that are countable. And here we see this very last plate appears to have a countable number of cells that can be visualized as distinct, what we call CFUs. And if we were to count the number of dilutions, we have 1 dilution, 2 dilutions, 3, 4, 5. There were 5 total dilutions. So it was 1 to 10 times 1 to 10 times 1 to 10 times 1 to 10 times 1 to 10, which equals a 1 to 100,000 dilution. That becomes our dilution factor, which we multiply by the number of cells here in order to arrive at our estimated original concentration of cells. Okay, number 18, log phase. Log phase is one of the parts of a normal cell growth cycle. So log phase is phase 2. It comes after lag phase. So after a culture is started in what we call a batch culture, so like we showed in the previous examples with those test tubes, if you were to take a 10 milliliter, vial or tube of sterilized medium, and you were to add some bacterium to it, this graph kind of shows the growth cycle that would occur in that culture. And remember, when we talk about growth, we're talking about the number of microbial cells. On the x-axis is time, on the y-axis is a log of number of bacteria. So initially, the bacterium are preparing for growth. This is an intense amount of activity where they're producing enzymes and cellular components and putting out their feelers to see what sort of nutrients and neighbors they might have. And they realize that it's time to go into max capacity growth. And that's what we see when we see log phase growth. And so log phase, again, is that sort of doubling every so much of time. And that's an exponential amount of growth, and so an exponential increase in population. But instead of seeing a steep curve right here, it's linear, and that's because this is a semi-log plot. So this is on a log on the y-axis and just a no log on the x-axis. So it appears linear to our eyes for this graph. After log phase, we enter into stationary phase where the number of deaths and new cells is in balance. And then finally death phase where the population decreases at a logarithmic rate. Okay, 19, UV light. Ultraviolet light is a type of light that is antimicrobial. They are wavelengths that are shorter than typical visible light. The most commonly used type is called UVC, And in lamps, that wavelength is 260 nanometers. Remember that wavelength is the distance, if you imagine it being a wave, it's the distance between one crest of that wave to the next, or one trough to the next, however you want to look at it. And what it does is it creates what we call pyrimidine dimers. Thymine is a type of pyrimidine that occurs in the DNA code. And normally these do not interact with neighboring ones. They only hydrogen bond across to the other nucleic acids. But in thymine dimers, they actually form bonds with a neighboring thymine. And this distorts the DNA and causes misreading of the DNA. So when replication occurs and it's time for the cell to make a new copy of DNA, it will misread this piece of DNA and make an incorrect copy. The same can happen when it's forming what we call mRNAs, messenger RNAs, which are another type of nucleic acid. We'll cover later in this course, but what you need to know is that interferes with the production of mRNA and DNA. This is the drawback of this method is it's less effective on liquids or growth media, and that's because UV light does not penetrate very well. Remember, we talked about how you can get skin cancer from UV light, but you don't get lung cancer from UV light. Okay, 20. Theory of endosymbiosis. This is the theory that mitochondria and chloroplasts arose when a prokaryotic cell was engulfed by a eukaryotic cell, and over time the two evolved to coexist under these circumstances, and eventually led to the development of chloroplasts and mitochondria, the descendants of that prokaryotic cell. There's a lot of evidence to back this up. The size, the DNA content, specifically what we call phylogenetics, and which composes of sequence similarities to determine relatedness. The ribosomes are similar, their membrane structures are similar, and because a lot of these structures like the ribosomes are similar, they also tend to have similar antibiotic sensitivities. Okay, 21, facultative anaerobe. Again, there is a lot that we can unpack here. I highly recommend that you put quite a bit of effort into studying this table 6.1. We have 5 main groups of microbes based upon their tolerances and ability to grow in the presence or absence of oxygen. We have the obligate aerobes, and so these ones are required to grow aerobically and they need oxygen. As you can imagine, that has to do with cellular respiration being their way for obtaining energy. The facultative anaerobes can do both, and as you will notice, they can appear as growing throughout this culture tube. This culture tube has a certain amount of oxygen diffusion in it if it's left undisturbed, and so what you'll see is towards the top is where most of the oxygen is, and when you reach the bottom it becomes very anaerobic, very low in the amount of oxygen present. And so facultative anaerobes have the ability to produce energy both aerobically and anaerobically. You will notice that there is more growth at the top, and now that we've had our discussion about microbial metabolism, you should kind of have an idea why. The reason for that being is cellular respiration produces a lot more energy than fermentation does. And so that's why you see a number of cells clumped here at the top. They do have, these two do have the presence of catalase and superoxide dismutase, and that allows them to handle toxic byproducts of oxygen, as we discussed with superoxide dismutase, that is superoxide, with catalase, that is hydrogen peroxide. We then have the obligate anaerobes, and they are the opposite of obligate aerobes. They cannot grow in the presence of oxygen, and they lack those enzymes in order to neutralize those oxygen forms. Then we have the aerotolerant anaerobes, which require a small amount, and they have the presence of one enzyme, the superoxide dismutase. I'm sorry, that I got microaerophiles confused. Aerotolerant anaerobes, they can only do their growth anaerobically, so they don't do cellular respiration. And that's why you see it kind of mixed evenly, and they don't have a bunch at the top. But they can tolerate some oxygen, and that's because they have superoxide dismutase. And lastly, we have the microaerophiles. These are a unique bunch because they kind of grow in the middle here. So they are able to do aerobic growth, but they need a very specific amount of oxygen. They require a lower amount of oxygen in order to survive. So they can't be exposed to normal levels of oxygen in the environment. And it can vary depending on which microaerophile, what sort of enzymes they do have to handle this. But you'll see them growing kind of in the middle here between the top and the bottom. And if I didn't say this already, oftentimes we call obligate aerobes and obligate anaerobes strict aerobes and strict anaerobes. 22. Quats. Quats are a type of detergent that disrupt the plasma membranes. So here we have acetylpyridinium, and it looks like this here. We have this, what we call a aliphatic or hydrophobic chain here, this hydrocarbon chain with no oxygens attached to it, which is very similar to what we see in the fatty acids in the bilipid membrane. And it's attached to this cyclic ring here with a nitrogen. But what's really important for you to know about this is not to memorize the chemical structures, but to know the components of it. So it has this hydrophobic region, and because this nitrogen is here with a positive charge, charged molecules are hydrophilic regions. The same over on this one, we have the positive charge here, and so this benzalkonium has a hydrophobic region and a hydrophilic region. So just like the phospholipids that have a hydrophilic and a hydrophobic region, they get in here and behave somewhat like them. However, the consequence is they disrupt this membrane, and that is how they are act as an antimicrobial. Okay, 23. Genus. This one should have been an easy one for you. It's the first part of the binomial nomenclature used to describe any life form. So all life forms have a genus and a species component to their name. The genus portion is written first. The first letter is always capitalized, unlike the species, which are entirely lowercase. And the whole name is italicized. So on an exam, you should be able to identify the proper convention for representing the genus and species of an organism. So here we have Escherichia coli, also known as E. coli, Salmonella, Enterica, Staphylococcus aureus, and yes, number 24, selective growth media. Okay, selective media allow some types of bacteria to grow, but not others. Differential media is not the same as selective media. It's very different. Differential media can allow multiple types of bacteria to grow, but there are differences in the way they behave or grow or look on that media that allows one to differentiate one species from another, or one strain or whatever. It allows you to differentiate between those different types. Media can be one or the other, or it can be both differential and selective. '25. Osmotic pressure. Okay, osmotic pressure is the pressure that can be exerted on a cell. It's one factor, along with temperature, pH, oxygen, and nutrients, that certain conditions are required for microbial growth. This proper osmotic pressure is usually an isotonic environment where there are equal concentrations of solutes inside and out of the cell. Remember, solutes are ions that are in solutions, such as sodium or chloride ions, any sort of salts. Sugars can be another one. What it does is it causes water to move inside or outside of the cell in order to try and restore a balance to those concentrations. So imagine in the cell, if there are more ions on the outside, or let's say more sugar molecules on the outside, then this cell is existing in a hypertonic solution. So it says NaCl 10%. So this NaCl 10% is a higher concentration of sodium chloride than what's present inside of this cell. And so water is able to move through this semi-permeable membrane without any problem because it's so small. So this water goes through simple diffusion outside of the cell until the concentration of salts is roughly the same, until it's in an isotonic condition. So this causes the inner membrane to shrink, and it can lead to plasmolysis, or disruption of this membrane. 26. Endospore. Okay, remember the endospore is a particular type of cell. The cell builds a essentially bomb shelter around it that makes it highly resilient and resistant to extreme conditions or environmental threats. And we can stain to observe these endospores using malachite green, and then malachite green is easily decolorized in cells that do not have an endospore structure, so we can just use simply water to decolorize, and then a counterstain of safranin to observe it. Of course, malachite green gives it a sort of a greenish color, and safranin provides this pinkish, reddish, purplish color sometimes as a background, as a differential stain. 27. Fimbriae. The Fimbriae are extracellular appendages used for attachment to cells and surfaces, and also can be used in the formation of biofilms. Number 28, inclusion. An inclusion, sometimes referred to as an inclusion body, are regions of a prokaryotic cell where nutrients and minerals are stored. These inclusions are particular for a particular type of nutrients. So you might have an inclusion body for the storage of nitrogen, phosphorus, carbon, things like that. (29) Alkylating agents. Oh, sorry, I jumped forward here. Okay, so alkylating agents are reagents that react with macromolecules in the cell. Often these are proteins, and when they react with them, they tend to just bind to them, and the reason why they're called alkylating is because they're actually alkyl groups. An example would be formaldehyde and glutaraldehyde. And so here's a protein and here's the formaldehyde, and they react forming this protein formaldehyde complex, and in doing so it changes the properties of this protein, and that's why they're found to be toxic. 30. Fermentation. Fermentation is a form of metabolism that does not use oxygen as the terminal electron acceptor. So on the top we can see cellular respiration, which does use oxygen in the final stage, and it accepts the electrons from our electron carriers. In this example we have NADH through the electron transport chain in order to generate ATP, and changes oxygen into water. In fermentation, after glycolysis, fermentation is used in the absence of oxygen. And basically in glycolysis, we produce 2 ATP, but we also produce NADH. And what fermentation does is it regenerates our NAD plus so that we can reuse it in order to generate more ATP in glycolysis. So the terminal electron acceptor in fermentation is some sort of organic molecule rather than oxygen. There are many different types of fermentation, and in this slide it represents several different byproducts of fermentation. In example A, there is no gas and no acid produced. In example B, a gas is produced, so this is probably CO2, but there is no acid produced from fermentation. In example C, we have no gas, but there is an acid that's produced. So in the process of finding a terminal electron acceptor, some sort of an acid is produced. This can be lactic acid, for example. And so lactobacillus could be C in this example. And then D, we have gas and an acid. So both an acid is produced and a gas, likely CO2. Saccharomyces, as another example, generates ethanol in fermentation and CO2 as well. Remember that pyruvate is the end of glycolysis, so that pyruvate is a 3-carbon molecule. And that 3-carbon molecule gets degraded into ethanol, which is a 2-carbon molecule, and its extra carbon is given off as CO2. All right, 31. Non-competitive inhibitor. So this has to do with enzymes. Enzymes need to be regulated so that they don't sort of run amok in the cell. The cell needs to control what sort of metabolic processes need to take place. In competitive inhibition, it does so by interfering directly with the active site. So an inhibitor molecule may bind directly to the active site, which is where the enzymatic process takes place for a given molecule. Non-competitive inhibitors bind to what's called an allosteric site. And so in this example here, we can see the allosteric site is distal. It's not located within the active site itself, but it does modify that active site so it is no longer active. Number 32, anabolism. Anabolism is a part of metabolism. It consists of chemical reactions that build up larger molecules from smaller molecules, and in the process consumes ATP, or the energy of ATP. This is the opposite of catabolism, the other side of metabolism, which breaks down larger molecules into smaller molecules and in the process can generate ATP energy. 33. Photoautotroph. Photoautotroph is a name for a microbe that determines where its energy source and carbon source comes from. In the example photoautotroph, its energy source is from the sun and its carbon source is inorganic, such as CO2. That means it's not combined as a hydrocarbon. And I specifically chose photoautotroph because I think it's the best example to help you memorize which part of the name has to do with energy source and which part of the name has to do with carbon source. With a name like chemoautotroph, it's a little bit more confusing. Does the first part or the second part have to do with energy? We know that photons are a type of energy, so it's very clear here that photo has to do with where the energy source is derived from. Auto means self in this example, so it's able to obtain its organic matter on its own, by itself, by using inorganic substrates. We also have the photoheterotrophs, which use the sun for energy, and organic compounds that others have made for its carbon sources. And some bacteria can do this. We also have the chemoautotrophs, which some bacteria can do as well. Chemo means that it gets its energy source from chemicals, and its carbon source is autotrophic, so that's inorganic. And then lastly, the chemoheterotrophs, which has its energy source from chemicals and its carbon source from something other. So something else has produced those organic compounds for it. And this is most bacteria and all of the fungi and all of the animals. So us as examples, we get, as we eat food, we get our energy sources from the chemicals in those food items that we consume, as well as our carbon sources. (34) NAD plus and NADH. These are two examples, or this is an example of an electron carrier. Electron carriers are those that pick up electrons, what we've discussed as in our example is glucose in glycolysis, and also in the TCA cycle. And its derivatives, such from NAD plus to NADH or FAD plus to FADH2, carry these electrons to the electron transport chain in order to conduct cellular respiration, which produces ATP and regenerates our positively charged NAD plus and FAD plus in order to repeat the cycle. And number 35, electron transport chain. Okay, the electron transport chain, there's quite a bit going on here, but essentially this occurs on the membrane, and through the use of several different proteins in this chain, it creates what's called an electrochemical gradient of protons. So those electrons are used to shuttle protons across this membrane. And then those protons are allowed to travel back across. They want to be in homeostasis, just like we saw with osmosis. Those protons want to even out in concentrations on both sides. And this is kind of similar to how, a battery works, where you have a charge difference between a negative terminal and a positive terminal. And those electrons want to travel to balance out that charge on both sides of the battery. So it's very similar like that, only this is membrane chemistry and not electrode chemistry. So in order to accomplish this, we want to use a very powerful terminal electron acceptor. And we have learned that oxygen loves electrons. So in this example, oxygen is the terminal electron acceptor. This occurs in the inner plasma membrane of prokaryotes. Or in eukaryotes, you should know it occurs on the inner mitochondrial membrane within eukaryotes. For each single glucose molecule, you will end up creating a total of 34 ATP with this process, and you will regenerate the NAD plus and the FAD. And in this diagram, we can see our inputs are our electron carriers, NADH, FADH2, and here are different bunch of proteins in this complex. NADH dehydrogenase, for example, hydrogen dehydrogenates recreates your NAD+. And these protein complexes take these electrons and exchange protons from one side to the other. And eventually these electrons have to get released somehow, and so here we see them combining with oxygen and hydrogen to make water. And the creation of ATP occurs. So here are the electrons flowing from one source or another. If you don't have oxygen present, of course this chain backs up and you need a backup method. Such as fermentation. And here is a wonderful sheet showing you how all of the different components of metabolism, the steps in metabolism break down. You should have this sheet pretty thoroughly memorized for the exam. You should know for each of these major steps, glycolysis, pyruvate oxidation, Krebs cycle, etc. What the major components that are made or lost are, and where it occurs. So glycolysis occurs in the cytoplasm, and then in eukaryotes, the rest of it takes place in the mitochondria. You should be able to add these up to determine the total amount of ATP or CO2 that's produced in each one of these. The NADH and FADH2 should equal 0 because when you regenerate things, they cancel out. Either in fermentation or in the electron transport chain, you should completely regenerate all of your NADH and FADH2 into NAD plus and FAD. You only get 2 ATPs though for ethanol and lactic acid fermentation, for example. So that wraps up our little game for using terms here and then trying to see how many descriptors you can think of to describe them and how much information you can recollect. The next about 10 slides are going to be covering the math problems I gave as examples in the book. the math problems that I give you on the exam are not going to be so wordy. These were just, ways to kind of make it a little bit funner, and so you could see the practicality of these math problems in all things microbiology. The first question is a bottled water analysis question, where a student wanted to evaluate the degree of microbial contamination of bottled water. So the student opens the bottled water and carefully aliquots or pipettes 0.1 milliliters of that water onto a nutrient agar plate. And the student incubates that plate and then comes back the following day after individual colonies can form. And remember, these colonies, or CFUs, represent one, or what's estimated to be roughly one microbe per dot, that then grows up into thousands and thousands of them so that it's visible with the naked eye. And that's called a colony forming unit. So the student comes back and counts 12 colony forming units, and now the student's job is to calculate the original concentration of bacteria in that bottled water. So this example, hopefully too, before we go through the answers here, you should try and attempt these on your own first, and then hopefully you can have a record of the mistakes that are made, and you can compare and contrast your calculations with mine and see, okay, what did I miss here and why did I miss it? And so when you get to the exam, you can say, oh, I forgot to do that one thing and I won't do it again here. So before you do this with me, try and do these questions on your own and then compare them to what I show you here. So what will throw many of you off is cellular concentration is listed as CFUs, or cells, per 1 milliliter. And if you look at this question, the student only pipetted 0.1 milliliters. So we have 1/10 of the amount of volume we should in order to list a cellular concentration in the correct units of cells, or CFUs, per mil. So since it's 1/10, it's just like a dilution factor. We just flip that around. We take the inverse of it, so it's 10 over 1, and we multiply that by what we got, which was 12. And so we'll see here, 12 divided by 0.1, or multiplied by 10, is 120 CFUs. I will accept either way you want to write it out, but I highly recommend getting familiar with scientific notation. Scientific notation has one value to the left and the rest of the values to the right of a decimal place, and then it's multiplied by 10 to whatever power to represent how many shifts that decimal place has one way or the other. So 10 to the second power means you shift this decimal place over 2 to the right. And so 2 to the right would give you 120 CFUs per mil. And it reads 1.2 times 10 to the second power CFU per milliliter of bottled water. So that wasn't too bad. Let's do the next one. Okay, so this is a tap water analysis. And the setup is a little bit different here. So the tap water is collected, and the way it's collected is 0.1 milliliters is added to a 9.9 milliliter tube of autoclaved, or sterilized water. And instead of 0.1 mils being plated, as seen in the bottled water analysis, a whole milliliter is plated. Your question is, if you come back the next day and you were to count 11 CFUs, what was the concentration in the tap water? And so we need to figure out our dilution, to figure out our dilution factor, and then we multiply that by the result to get the original concentration. So our dilution is the reverse of our our dilution factor is the reverse of our dilution. Our dilution, we took 0.1 mils and we added it to 9.9 mils, or we added it to a total of 10 mils. Okay, this will throw a lot of students off. Remember, it's always the total. It's not what you started with. It's the final amount of water after adding everything together and mixing it up. So we have a 10 over 0.1, which is equal to 100, or 10 to the second. Our final concentration is, you just take your result, which is our 11 CFUs per mil, and which is 1.1 times 10 to the 1 CFU per mil, and we multiply that by our dilution factor, 10 to the second, and that gives us 1.1 times 10 to the third CFU per milliliters of tap water. This is one of the reasons why I highly recommend you getting used to writing in scientific notation, although it won't be required for you to write it out that way for the exams. It's very easy to do calculations involving large numbers, because these exponents, so we had our 1.1 times 10 to the 1, or just 10 CFUs per mil. If we multiply that by our dilution factor, which is 10 to the second, you just add these two together. So 10 to the first plus 10 to the second is equal 10 to the third. So that becomes 1.1 times 10 to the third. It's pretty simple. And if you want to convert this to just a whole number without the scientific notation, 10 to the third means, again, you move that decimal place 3 times over. So 1, 2, 3, and that leaves you with 1,100 cells. Okay, next up is the urinary analysis. So here we have a patient who arrives at the hospital, and the patient presents symptoms of dysuria, which is pain during urination. So the doctor thinks that the patient could have a bladder infection, and orders a urine bacterial cell count test. And so what this does is it counts the number of bacteria found in the urine. This will require a dilution, because urine, it's not a, contrary to myths, it's not sterile. It picks up bacteria on its way out. And so a certain amount of bacteria, if there's a high enough load of microbes, it's indicative of a bladder infection. So a sample containing more than 1,000 cells per milliliter of urine is indicative of a bladder infection. So what happens is the urine is collected in a sterile container, and then 0.1 mils of that urine is diluted into 0.9 mils of water in this example. And so what should the total be? That's right, the total should be 10 mils. So we have 0.1 and 10 mils. Then we do some microscopy counts. So we take a hemocytometer, which remember has the grid on it that allows you to count the number of cells in a given volume. And we examine 0.01 mils of urine and count the number of cells. The number of cells counted were 16. So now the question is, what is the original cell concentration, and is it indicative of a bladder infection? Now this question might seem a little overwhelming. You know, there's a lot of 0.1 mils, 0.01 mils that are being used. How in the world are we going to get to this original concentration, and where do we start? And remember, you're always going to take the number of dilutions you have, you're going to turn it into a dilution factor, and then you're going to calculate how many cells you have per milliliter, not 0.01 milliliters, not per liter. Concentration is going to be per milliliters. So if you're given less than a milliliter, then you can just include that in your dilution factors, and we'll see how that plays out here. So again, a dilution factor is the reciprocal of the dilution, or the opposite if you flip it upside down. And we had 1 mil of total volume. It was 0.9 mils of water plus 0.1 mils of sample, and 0.1 mils added. So our dilution factor is 10 because our dilution was 0.1 to 1. So 1 divided by 0.1 is 10. And our final concentration of cells, which is the number of cells counted divided by the volume examined. So in the case of that first plate count, the volume examined was 0.1 mils. In the case of this hemocytometer, the volume examined was 0.01 mils. And the person counted 16 cells, so 16 divided by 0.01 tells us the number of cells per mil in that final concentration. All you really do here is you actually end up just timesing by 100 is the same as dividing by 0.01. So in a way it's another dilution factor. But if you would like to keep it simple, you can just take cells counted or CFUs counted divided by the volume examined there. So we have 1.6 times 10 to the third cells per mil. Then we have our calculation to get back to the original concentration, which is our final concentration, or what our examined concentration was, multiplied by whatever dilution factors we have, equals the original concentration. So let's take our numbers and plug it in here. Final concentration 1.6 times 10 to the third cells per mil, and our dilution factor was 10. Our final answer, if we add up the 10 to the third and 10, or multiply however you want to do it, it's 1.6 times 10 to the fourth. And this answer is a concentration, so it's cells per milliliter of urine. And remember, this was a two-part question. It also asked, what is the urinary cell concentration, and is this indicative of a bladder infection? So it had to be more than 1,000, and as we can see, 10 to the fourth means we move this decimal place 6 times over. So once on the other side of 6, and 3 more to add 3 more zeros is 16,000 cells per mil. So the answer is yes, this concentration of bacteria in urine is indicative of a bladder infection. Next up is wastewater treatment. So a wastewater treatment plant needs to check the number of coliforms. Coliforms are a type of bacterium that can grow in the intestines of animals. And so before a wastewater treatment plant will release its water back into the environment, it needs to meet certain thresholds of the number of coliforms present in that processed water. So what they ended up doing is they filtered the water using a 0.2 micron filter, and so that should catch all of the microbes onto that filter. And then they stick that filter directly onto an agar plate. So if there were any microbes that were in that water sample, it should end up on that agar plate. And that agar plate is an eosin methylene blue plate, or EMB. And this plate actually selects specifically for, it can select specifically for coliforms. It's used to identify coliforms anyway. So it's a selective plate. And essentially, in order to do this properly, that water sample had to be diluted, because you can imagine there's going to be potentially a lot of coliforms in that water sample. So before filtering it, they dilute it in 1 mils into 9 mils, so that's a 1 to 10 dilution. And then that 1 mil of this dilution was then diluted again in a 1 to 10 solution. So we have 2 dilutions. And then one of the mils was filtered through the filter and placed on the EMB plate. So we examined 1 milliliter, and the people at the plant counted 20 CFUs. So that's 20 CFUs per milliliter. And so the acceptable limit is 10 bacterial cells per mil of effluent, so processed water. And now we need to determine what the original cell concentration was of the water sample, and is it low enough that we can release this water into the environment. Okay, so remember I said we need to first find our dilution factors. We need to find what our final concentration was, and that's the cells counted per volume examined, and that needs to be in cells or CFUs per milliliter. And then we multiply the two together to get our original concentration. So here we had water that had to be diluted first because we were afraid we would get too many bacteria. So we did two serial dilutions, a 1 to 10 and a 1 to 10, which is a reciprocal of 10 over 1 and 10 over 1. And when you do serial dilutions, you just times one dilution factor after another to get your final dilution factor. So this is 10 times 10 equals 100, or you can write that as 10 to the second power. And we counted 20 CFUs, and remember that we only filtered 1 milliliter. So we counted 20 CFUs per milliliter, which is 2 times 10 CFUs per mil, meaning that we shift that decimal place one time over because it's 10 instead of 10 to the second, which would be shifting the decimal place twice. We multiplied the two of those together, so our concentration is 2 times 10 CFU per mil. Our dilution factor was 10 to the second, and so that's 2 times 10 to the third cells or CFUs per milliliter. And we wanted to make sure that we had less than 10 bacterial cells per milliliter. And this concentration is way too high. It's not low enough to be released into the environment, so this water needs to be further processed before releasing it. Okay, I have another one that I stuck in here, and this is genetic engineering of an aerotolerant anaerobe. And we have a graduate student who's trying to see the effects of the gene that encodes catalase. So this student took this gene and introduced this gene into an aerotolerant anaerobe. So remember, these beasties, they don't do aerobic respiration, but they can tolerate some oxygen. And so this catalase gene was introduced into this microbial species, this bacterium, and then they wanted to see how it would grow in aerobic conditions. So the original strain and the one with the catalase were both aerobically cultured overnight. So they were just grown on a normal shaker, which gets exposed to the air. And in the next day, 0.01 milliliters of the culture were diluted into 9.99 milliliters of sterile liquid medium. It was mixed, and a volume of 0.01 milliliters was observed under a hemocytometer. A total of 13 cells were counted for the catalase mutant. What was the original concentration? And the original strain, so the one without catalase, had 1.3 times 10 to the 5th cells per mil. Did this gene help grow under aerobic conditions? Okay, so again, we want the hint here is you need to have your answer in cells per mil, not cells 0.01 mil. To begin with, I recommend always kind of finding your dilution factor, and we added 0.01 to a total of 10 milliliters, and so that's 10 over 0.01, which is equal to 1,000, or 10 to the third for dilution factor. And our final concentration was 13 cells in 0.01 milliliters of volume examined. So 13 divided by 0.01 is 1.3 times 10 to the third cells per mil. We multiplied those two values together 1.3 times 10 to the third cells per mil times our dilution factor 10 to the third, and our final concentration is 1.3 times 10 to the sixth cells per mil. So did that help this organism grow in aerobic conditions? It does appear so. We had 10 times more cells growing with the catalase than without the catalase gene. Okay, our final question is a brewing question. So a brewmaster wants to ensure they have enough yeast cells to brew a batch of beer. And a sufficient number of yeast cells, which is called the pitch rate, is 200 billion cells. So we could write that out as 2 with a bunch of zeros. In other words, 11 zeros, or we could just simply use scientific notation. Let's just write it out like that. So this is how many times you'd have to shift the decimal place to the right. So you'd have to move it over 11 times, and you get 11 zeros to make it. So it's 2 times 10 to the 11th. So to calculate the number of cells she dilutes the sample down and counts the cells under a microscope on a hemocytometer. There are 5 1 to 10 serial dilutions that were done. So the 1 ml culture was added to 9 milliliters of water, and then that sample was mixed and diluted again. So this was done 5 times. And the last dilution cell counts were done. So on average, I suppose this particular counting happened several times to make sure it was reproducible, but on average there were 22 cells were counted per 0.01 milliliters, or that's also can be called 10 microliters. There's 1,000 microliter per milliliter. So we'll stick to the 0.01 milliliters though. And so if she adds 1 liter, and there are 1,000 milliliters in a liter, she adds a liter of this original culture, how many yeast cells will go into the wort, and is this a sufficient yeast pitch rate? So this is just building upon what we've done in the past. We're going to find the dilution factors. We're going to find the number, the final concentration in cells counted per volume examined. Multiply the two together to get the volume used. Let's look at our dilution factors first. Remember, this was 1 ml added to 9 ml of water, so that's a 1 to 10 dilution, and it was 5 times. So the reciprocal of that is 10 over 1, and we know that in order to figure out the final dilution factor, we just multiply all of our reciprocals of all of our dilutions. So since there was 5 here, we do it 5 times. 10, 10, 10, 10, 10. So that's 100,000, or we can just simply move the decimal place over 5 times and say it's 1 times 10 to the 5th or 10 to the 5th, if you simplify that there. Next step, let's find the final concentration of our cells. So we counted 22 yeast cells, and we examined 10 microliters, or 0.01 milliliters. So that gives us a nice answer, 2.2 times 10 to the third. And let's multiply the two together. 2.2 times 10 to the third yeast cells per mil times our dilution factor, 10 to the 5th, equals 2.2 times the addition of these two exponents. 10 to the third times 10 to the 5th equals 10 to the 8th. Remember, you always add the exponent. Don't multiply these together. It's an addition. It's not 10 to the 15, it's 10 to the 8th cells. 10 times 10 is 100, right? 100 times 100 is 10,000. So it's 2.2 times 10 to the 8th. If this confuses you, you feel free to write it out in the long form. So the yeast cells added is going to be equal to the cell concentration times the volume used, naturally. So if we use 2 mils of this, then it would be 2 times this answer here, and that would be 4.4 times 10 to the 8th, right? If you had, I don't know, if you had 2 billion cells and you per mil and you added 2 mils, then it would be 4 billion cells, right? So we said in the problem that we were going to add 1 liter, which is 1,000 milliliters. So let's take our concentration, 2.2 times 10 to the 8th yeast cells per mil, multiply it by 1,000, or 10 to the 3rd, and that, if we add those two numbers together, we get 2.2 times 10 to the 11th yeast cells. And we remember here we needed 2 times 10 to the 11th yeast cells to have a sufficient pitch. So we have plenty of extra yeast cells, and we did meet our quota. That will do it for the math questions. I hope you found them helpful and also a little bit fun. For the exam, you'll have something very similar to these questions, probably a little bit less wordy. So you should feel comfortable in finding dilution factors, and that shouldn't matter if it's a single dilution or multiple dilutions. You should be able to find out your final concentration in cells or CFUs per mil, and you should be able to multiply your dilution factors and your cell concentration to get your original cell concentration, and compare that to the required answer to see if it's a positive test, a negative test, if you're meeting or not meeting a threshold, just sort of basic logic on that end. So thank you so much. I hope this helps for the exam. Please reach out if you have any further questions. I'll be happy to help. Take care.